introduce friend and colleague Jonathan Pruitt from UCSB. He studied marine biology as an undergraduate at University of South Florida, studied animal behavior for his PhD at University of Tennessee, Knoxville with Susan Reichart, and then did postdoc work at Davis with Andy C. and Jay Stakowicz. He had a position and was tenured at the University of Pittsburgh, and then joined UCSB's EEB department in 2016. Jonathan is a star, not a rising star, a star in the field of animal personality. He is a social spider enthusiast, but also studies other invertebrates as well, including some marine invertebrates these days. And what's fascinating about his work is it's integrative and interdisciplinary, and then crosses levels of biological organization, looking at the consequences of personality, in this case for extinction as well, and population persistence. He's a thought leader, a well sought after speaker, and publishes a lot and all in the best journals. In 2015, he was named one of the Brilliant 10 Popular Science Magazine's top 10 minds in science, engineering, or mathematics under 40. Please join me in welcoming Jonathan for today's talk.
Thank you very much for the kind introduction, and thank you all for coming. I guess I am mic'd. Yeah, I've always admired your department, and so I'm pretty excited to share some of the work that I've done with you guys, and I hope to not bore you for about the next 50 minutes. And as Dan mentioned, and as my first slide kind of gives away immediately, I study social spiders, which cooperate like wasps or bees to make giant colonies together. And this is a picture of one of these natural wonders of what these animals are capable of. And this is a singleton web containing more than 50,000 individual spiders from 40 or 50 different species that make a shared architecture together, like a silken reef inhabited entirely of predators. And those are two normal-sized people. This web went for 200 yards and engulfed several oak trees. So what maintains diversity? Pretty much all of ecology and evolutionary biology is to some degree preoccupied with diversity. And so maybe you're interested in what allows different male lizards to maintain independent reproductive strategies in the face of competition. Or maybe you're a metapopulation biologist and you're interested in how is it that different subpopulations can maintain independent evolutionary trajectories in the face of gene flow. Or maybe you're a community ecologist and you're interested in what allows so many species to occur in these biodiversity hotspots like neotropical rainforests or Indo-Pacific reefs on finite resources in spite of all that competition. But then there's the sort of cynical counterpoint to any of this that we're going to focus on today, and that is, why do we care? What does a phenotypically diverse population of lizards do that's special or unique that we should care about? Or what is a diverse metapopulation with independent trajectories within it, whatever the heck that could possibly mean? What does that do functionally in aggregate that we could care about? Or what is the functional significance of having a speciose system like a neotropical rainforest? Why don't we just mow it all down and make it a giant monoculture of some super species? And I answer these questions or address these questions in animal behavior under the guise of behavioral syndromes or maybe animal personality, which is just defined as temporally consistent individual differences in behavior that manifest across time, situation, ecological context. And so it kind of sounds a little fuzzy. So this is part of the talk where I ask, how many of you have ever had a dog? Raise your hand. I'm not going to call on you. It's not one of those talks. Or seen a dog or know what a dog is. Got it. All right. So anyone who's had a bunch of dogs, and we'll say that you're a corgi enthusiast, and you allow your corgi to have a litter, and you become familiar with each of these little puppies from the exact same female, right? And you would say, because you know them so well, that they differ in their behavior, little personalities, right? Some are more aggressive, some are more active, some are more needy, some are more sedentary. And because you know these animals quite well, you can establish a sense of their individual differences in behavior. These happen in all domesticated populations of animal, and they don't just occur in domesticated populations or in people. They occur across the animal kingdom, even among clonal individuals, and they have enormous consequences for how individuals live their lives. So maybe you're a stickleback. Like on the left-hand side here, and individuals might differ in, say, their activity level. If you're an active stickleback, you move around more in your environment, you capture more food, you go to a larger body size, you have more eggs, maybe you have other advantages as well, but generally speaking, it's good to be an active individual, right? So why aren't all individuals active? Well, that's because the most active individuals that grow the most quickly also are more apparent to their predators and are more likely to succumb to predation, right? And it's the sedentary individuals that are slow growing and might not reach such a large body size that are safer, right? So a growth survivorship trade-off can be determined based on an individual's personality type. On the right-hand side here, we'll talk about funnel web spiders, and this work is done by my undergraduates, where females differ in their level of aggressiveness. Generally speaking, as a female spider, it's good to be aggressive. You get tons of food, you get the best territories, you defend that territory from intrusion by conspecifics, you grow to a large body size, you have an awesome egg case, huge, filled with eggs. So why aren't all females wantonly aggressive? And the problem is that the same females that have the best territories, that have the largest body size, that have the most eggs, also suffer from a maladaptively high incidence of precopulatory sexual cannibalism. And what that means is that these females end up killing all of their would-be suitors and dying. Virgin spinster spiders having never actually made it at all, right? So all of those eggs, that awesome territory, is basically irrelevant because she never even had a successful mating opportunity, right? So she kills all of her would-be suitors, effectively sterilized by her high levels of aggressiveness. And that's one of the elements about personalities. You can't just turn it off when it's convenient. You have some consistency to how you behave. All right, so there's two key questions sort of floating around in animal personality. I guess this is actually kind of dated in this way. There's lots of questions floating around, but two general spheres that we'll go over today. And that is just basically, how does an individual's personality type influence its performance in different contrasting environments, its survival, its reproductive output, things like that. And then because I grew up in a department where behavior was sort of like the, can I say redheaded stepchild? Can I say that? The redheaded stepchild in the department, and sort of viewed as like, like the soft-headed element of the department. I oftentimes found myself pondering how individual variation in behavior influenced higher order ecological processes that general ecologists might care about. For instance, the stability of populations, intrinsic growth rate, carrying capacity, species interactions, et cetera, et cetera. And that is what we're going to perseverate on today, assuming that some of you are not inculcated in the world of behavioral ecology. All right, so social spiders. You saw the first slide of social spiders. Now I'm showing you lots of other pictures of cool social spiders. How many of you before today even knew that social spiders were a thing on planet Earth? Raise your hand. Pretty good. Okay, so like 60 or 70% of the room. But most normal people, not you guys, actually had no idea that social spiders even exist. And that's because out of the 48,000 species of described spider on Earth, only about 25 are social. And new species of spider are being identified all the time and described, but not a whole lot of new social species. And so they're not an incredibly ecologically significant or speciose group, but they have a lot of traits that make them highly amenable to study that I'll outline in just a moment. So because most people don't know anything about social spiders, we're going to just do some spider biology 101 and group living. So on the left-hand side here, we have colonial spiders, which are a more common phenotype. And those are, there are about maybe 200 different species. And the exemplar here is Sotophora from Renomafauna, Madagascar. So colonial spiders, they make these giant aggregations. But within that giant aggregation are these little subwebs that are each occupied by one female. And so if another female intrudes on a focal female's web in order to, say, capture prey, they're very likely to engage in agonistic interactions. They're not very affiliated with each other. They maintain these territories, and they don't spend much time grooming each other or assisting each other beyond this just general coarse aggregation. And on the right-hand side here, we have social spiders, which that's only about 25 species on Earth. And the exemplar here is Stegodyphus saracenorum from Andhra Pradesh, southern India. And within this giant colony here, there are these little fist-sized nests. And each of those fist-sized nests will contain anywhere from about 50 to about 2,000 spiders that are nestled right up against each other. And they cooperate with in a variety of tasks. But the most important thing here is that they're very tolerant of conspecifics. So first off, all the spiders we're going to talk about today are females. And that's because most social spiders are female. Social spiders have a highly female biased primary sex ratio. So if you just crush the embryos as soon as they're fertilized, they're between 8 and 10 times as many female embryos as male embryos. And that female biased sex ratio persists throughout their life cycle. Females cooperate together in a variety of tasks. They create these giant silken weaves together. So they maintain these giant webs. They capture prey together like a pride of lions. So although although the social spiders themselves might be like 5 millimeters long, they can capture morpho butterflies or lubber grasshoppers or small sunbirds or bats. I think that's quite impressive. I'm waiting for the day that I find a little human carcass inside of one of my colonies. Hasn't happened yet, but they are capable of subduing pretty impressive prey. And then the last thing that they ever do is they engage in suicidal alloparental care. So they will capture prey for their offspring, and like all spiders, they vomit up the contents of their stomach to liquefy their prey, and then they slurp that vomit back up, and they vomit that into the mouths of their offspring, like a mama bird would do. But then just before these females die, and they all die in unison as a cohort, they liquefy their own bodies via programmed cell death, and then are ripped apart and consumed by their prey. And we actually know now that while they're vomiting up the contents of their stomach to feed their prey early on, that decomposition process has already started. So they're actually vomiting up elements of their own viscera into their offspring's mouth for weeks before they finally perish. Right. And so it's also thought that these are foraging societies. So the reason that sociality evolves is because of the ability to subdue larger and more profitable prey size classes that are otherwise unobtainable for a singleton spider. And then finally, males don't really do a lot in these societies. Once they near maturity, they basically stop eating. They don't produce any silk. They just mate with a few females, maybe even lots of females, and then they perish. It's really females that are the movers and shakers socially inside of these societies. Males are just sort of vectors for sperm. All right, so Anolosmus studiosus is the social spider we're going to talk about today, and that is the one species of social spider on planet Earth that isn't tropical. They're almost all tropical, except for this species, which occurs in Appalachia along riparian systems throughout the southeast United States. But they have a huge geographic range. This species occurs from Maine to Patagonia and Argentina, but the multi-female colonies only occur in Appalachia, and they occur in temperate climates throughout that distribution. Females exhibit one of two temporally consistent personality types. Females are either very, very aggressive or incredibly docile. And so we'll talk about what that is in just a moment. How do you determine a spider's personality type? It doesn't have a lot of opportunities to self-report. And so what you do is you take two females of unknown personality type, you put them together in a box, and you leave them for 24 hours. If after 24 hours they've cuddled together in the same corner, we score them as docile because they are docile of conspecifics. They're willing to nestle right up against a could-be competitor. If they settle in opposite or adjacent corners, we score them preliminarily as aggressive because they demand space from conspecifics. This is a preliminary assessment. And the reason that is because one of these two females might actually be docile and tend to aggregate with the other female, but is repelled by the second female's aggressiveness. So every single female has to be retested with a known docile individual to identify how much space she demands, her inter-individual distance when she's with a cuddly conspecific. I've never said that alliteration before, but there you are. All right, so this is highly repeatable. Individuals that are docile today tend to be docile in the future. Individuals that are aggressive today tend to be aggressive in the future. There's a heritability to this trait. If your parents were aggressive, you're more likely to be aggressive. If your parents were docile, you're more likely to be docile. And that's confirmed by mid-parent on offspring and paternal half-sib breeding designs. And we're not just talking about how much space a female demands here. These more aggressive females in these stage interactions are more likely to attack prey more rapidly. They're more likely to waste food. They're more likely to attack their predators. They're more likely to kill their mates. This quick and dirty assay is correlated with a constellation of phenotypes or elements of a female's life, and we use this as a quick diagnostic assay to try to capture a constellation of traits of interest, a Pleiades. All right. So colonies contain a mixture of aggressive and docile females. So we just have a frequency histogram here of colonies assayed in a single focal population. All the way to the right would be a colony of all aggressive females. All the way to the left would be a colony of all docile. And all I want you to get out of this is that colonies vary in their relative representation of docile versus aggressive females. And so that's the jumping off point for this whole talk. What does a colony of all aggressive females do that's different from an all docile colony or a mixed composition, and which colonies are favored and under what circumstances? So early on, I thought that this aggressive phenotype might be a socially parasitic phenotype, right? They demand space, they're super aggressive, they're more likely to cannibalize conspecifics, and so I thought they would depress colony performance. And we ruled that out very quickly early on. So we're looking at a figure of colonies reared in a common garden of either all aggressive females, all docile females, or a mixture of 50-50, with the prediction that adding aggressive females to a colony would reduce the colony's performance. The y-axis here is just a female's standardized reproductive success. It's her egg case mass divided by her pre-mating body mass. And what I want you to get out of this is that colonies of mixed phenotypes are producing heavier egg cases. So there's some sort of a positive advantage of having within-group behavioral diversity, social heterosis, one might call it, and these mixed compositions are outperforming either monotypic composition. You might say here, though, this doesn't really get to the point, though, right? So maybe docile females within this mixed society are doing horribly, right? And aggressive females are doing great because they're parasitizing the affiliative positive effects of these docile females. So if we just take that mixed composition and decompose it and compare the egg cases produced by docile and aggressive females within those mixed compositions, we see that they're doing equivalently in terms of their performance. So both docile and aggressive females produce heavier egg cases with more eggs than they would in monotypic societies of either type. So there are positive effects of within-group behavioral diversity. It doesn't appear like one's just exploiting the other. So the point of my dissertation, and this is basically walking through about half this talk is from my dissertation, about half of it's from my work at Pitt. I wanted to sort of walk my way through all the challenges of being a spider and then seeing how manipulating these ratios would influence all these outcomes. And so what do you have to do as a spider? We have to capture prey. You have to defend your colony from intrusion by heterospecifics. You have to repair your web because it rains every so often. It's a three-dimensional web, so you got to repair that. And then the last thing is you have to rear your offspring, and that could be troublesome as well. And it basically turns out that I never really left colony defense. So these silken reeves, in addition to harboring the social spiders themselves, will serve as hosts to now we know more than 100 different species of foreign spider that are normally solitary species that are attracted to these social spiders' webs, infiltrate them, and exploit them, we assume, to some degree. And so notably, each and every year, between 60 and 90% of colonies go extinct. That's where everybody dies, nobody escapes, death, 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 little spider carcasses blowing in the wind. And so we wanted to assess to what degree do these foreign spiders influence the probability of the foundational species going extinct? Or do they influence the performance of their foundation species? So silken reeves. So we know that, so this is now outdated. We now know there's more than 15 different families that co-occur in these webs. There are more than 30 different genera. But the point is, there's a lot of stuff that occurs in these colonies. So first off, we know that a lot of these foreign spiders could be kleptoparasites. So we know that they steal prey from other spiders, but normally solitary spiders. We assume they're probably doing something similar and bad to these social spiders. But most of these foreign spiders, we have no idea what they're doing, individually or in aggregate to the performance of the social spiders themselves. But we know it's probably not good, because they're anywhere from 5 to 50 times larger than the social spiders themselves, and they're really aggressive polyphagic predators. So we assume they can't be helping these social spiders. And so first question for today is, how might the phenotypic composition of a social group influence your probability of interacting with these heterospecifics, whether they're helpful or whether they hurt the host species? And so we decompose that into two smaller questions. What might be the effects of foreign spider diversity on colony performance if you have a diverse suite of foreign araniofauna or a very limited number of species? And then secondarily, whether or not these foreign spiders help or hurt the focal colony. Does the composition of the focal colony change the diversity or prevalence of these foreign spiders in the web? We focus on diversity for a reason that will become totally transparent in about 2 slides. I'm not just bamboozling you. It'll become clear in just a moment. So first we wanted to see whether or not we could predict colony survival as a function of the foreign spider community that infiltrates the colonies. So we went out into the field, we dissected open 55 colonies, we slit them open with a razor blade, we ID all of the foreign spiders to family. Is it a jumping spider, or an orb weaver, or a sac spider, or a pirate spider, or whatever, and then we just leave the colony alone. Because it's basically a weird filter feeder that happens to just be in the field and occupied by spiders. We just leave it there, and then we come back 4 months later to see whether or not the colony's still around, and then we predict whether or not we can predict whether or not that colony's still there or not at the end. So we use the total abundance of foreign spiders, the abundance of each kind of family, jumping spider, whatever, and then the total number of families, which is our metric of diversity here, and the response variable is just colony persistence. And I'm going to show you the single best model here for a moment. So what predicts colony persistence? First things first, the more different kinds of foreign spider families you have in your colony, the more likely you are to die. So if you have 2 or 3 different foreign spider families inside of your colony, a low diversity, you're very likely to live. If you have 5 or 6 or 7, a diverse community, it's very likely to die, right? So here, biodiversity, the representation of different kinds of foreign spider families, actually drives the extinction of the foundation species and the subsequent disarticulation of the community, right? Diversity isn't always such a great thing. it's an assemblage of co-infecting agents like this anyway. Secondarily, the number of agilinids that are in the colony are negatively associated with colony performance or persistence. And that's not that shocking. Agilinids are about 25 to 50 times larger than the social spiders themselves. They can run 50 centimeters a second. They can subdue 50 plus prey items in a single bout. This is a thing of nightmares to my social spider, and the more that you have these things inside of your colony, the more likely that you and all of your sister cousins are going to die together as a unit. And then finally, and this is the weirdest one, is that the number of pirate spiders inside of the web is actually positively associated with colony persistence. And pirate spiders are really weird spiders. They're famous for their araneophasic tendencies, and that means they specialize at eating other spiders. So if you have a spider-eating spider inside of your social spider colony, you're more likely to live. Which doesn't make a lot of sense if you think that it's eating the social spiders, but it might make sense if it's eating the other spiders inside of the colony, right? So the predator of thine predator is thine friend, perhaps. And if you buy that argument, then we'd be talking about a trophic cascade occurring among a community of of predators living at a shared biomaterial suspended in a silken pendant over some crystal clear river in Appalachia. And how could that not be impressive? All right, so that's nice. So we know that in general, though, in aggregate, foreign spiders bad. Occasionally good with the pirate spiders, but on average bad. So does the composition of the society change whether or not you're going to have many of those foreign spiders or just a few of them? So we went up to naturally occurring colonies. We took 50 of them. We brought them into lab now. And so we can actually look at these things under a microscope. We count the number of social spiders inside of the colony. We determine whether or not they're docile or aggressive. And they ID all foreign spiders to the genus because it's easier to do when you've got a microscope and they're dead. All right, so can we predict the number of foreign spiders inside of the colonies? So inquiline is a weird word that just showed up. All inquiline means is that you're an animal that lives in the domicile of a social animal. That is not conspecific. So these foreign spiders are inquilines of the social spiders. And so the y-axis here is the genus richness, the number of genera, and along the x-axis is colony size. And not that shockingly to anyone who's ever looked at anything like this, oh gee, what do you know? Larger habitats that have more social spiders tend to have a more diverse assemblage of foreign spiders. Not shocking that larger habitat patches would have more diverse community of foreign spiders. The key question is whether or not the composition of the group is associated with this representation of foreign spiders. X-axis is now the proportion of the colony that's aggressive, so all the way to 1 would be 100% aggressive, all the way to 0 would be all docile, and the y-axis is the same as before. And this is the crappiest, most anemic Diet Coke data in the whole talk. I apologize, but bear with me. Presto changeo, there it is. All right, so there's a strongly significant negative association between the representation of the aggressive phenotype and the diversity of foreign spiders that you have in your colony. So if you have a colony of all aggressive individuals, maybe you'll have like 3 different genera. And if you're a colony of all docile individuals, maybe you'll have a community of 5 different genera. And that actually seems pretty pathetic, right? It looks like a buckshot on that data set. But if you harken back to the relationship between foreign spider diversity and survival before, which is incredibly nonlinear, the difference between 3 and 5 might look pathetic here, but that's the difference between almost certainly going to live and almost certainly going to die in that other analysis. And so although this is sort of a weak, middling statistical association, I argue that it might actually be really important biologically. And I have more data to fortify that notion or evaluate that notion subsequently in this talk. All right, so are aggressive females colony defenders? So we know that aggressive females are better at capturing prey in lab and that they'll share that prey with docile females. We know from here that aggressive females are negatively associated with these foreign spiders and that these foreign spiders are on average bad, right? So maybe they're acting as some sort of colony defender and maybe they're really valuable for the performance of these societies. So let's flip this whole system around on its head now. Let's pretend that we're not focusing on social spiders any longer. Now we're focusing on the foreign spiders that infiltrate these colonies. And if we think about these patches of social spiders as sort of unique habitat patches that differ in their phenotypic composition, some patches might be more profitable than others to infiltrate if you're a social parasite. So if we're all social parasites and we're thinking about infiltrating some social spider colony, which one would you prefer to infiltrate? The colony of all aggressive females with high activity levels that are very likely to attack you? Or the colony of all docile aggregative lambs that, it's very clear where I would want to go. I'd want to exploit the colony of all docile females. And presumably, colonies of all docile females are very susceptible to exploitation by these heterospecifics. So what do? We want to track the performance of these heterospecifics when they occupy colonies of various compositions. And we use Agelonopsis emertoni, which is one of those grass spiders that we know is very bad for colony performance, and Lerinioides cornutus, which is an orb weaver that basically just hides inside of the colonies during the day and then makes an architecturally unique Charlotte's Web, orb web at night. And then when it catches food, it wanders back into the colony. So we picked 2 different species that are large, easy to mark, that occur in greater than 50% of the colonies that we census, and that have a difference in their intimacy of the association with the focal species. Agelonopsis emertoni basically lives directly within the retreat of the social spiders themselves, and then Lerinioides cornutus is sort of just a peripheral species that builds its own architecturally unique web. So how might their performance differ based on the representation of docile versus aggressive females? What do we predict? Docile colonies will be more profitable habitats because they're just less defended, right? And presumably these docile females are going to suffer greater costs when they're infiltrated. So I made artificially reconstituted colonies and deployed them into the field of either all aggressive females, a mixture of types, or just docile females. And then before I put them out there, I apply 1 of 3 treatment groups. I either just throw in a control glass bead, which presumably is not exploiting the social spiders, right? Or I throw in an Agelonopsis emertoni or Lerinioides cornutus, and then I track the performance of both these inquilines in terms of their change in mass and how the social spiders themselves are doing in terms of their egg case masses. I also released some of these foreign spiders by themselves into the population to see how they would do if they were outside of the social spider colonies themselves. And then I tracked the performance over 6 weeks in the field. So we're going to be looking at the relative change in mass of Agelonopsis emertoni when it's in a colony of all aggressive, 50-50, all docile, or when it's outside of the host colony. And consistent with our hypothesis, yes, if you're in Agelonopsis emertoni, you're gaining way more mass if you've infiltrated a patch of all docile lambs. You're always doing it at least as well as you would in isolation, but you're really only gaining more mass when you've infiltrated a docile group. Lerinioides cornutus, same axes as before. Same story. So they're really, and there's very little variability here, but and it's not because the colonies went extinct. So this species is gaining a ton more mass when it's infiltrated a society of all docile females, presumably because they're lacking defenders. It's always gaining at least as much mass as it would in isolation, but it's really only profitable in terms of mass gain for this species to infiltrate docile groups, presumably because they're easier to hurt, to steal from. So how are the social spiders themselves doing? So on the left-hand panel here would be a colony of all aggressive. The middle panels would be 50-50. Right-hand panels would be a colony of all dociles. And then we'll throw up 1 treatment group at a time. The y-axis is just the relative reproductive success of females occupying each of those compositions and treatments. So in the absence of foreign spiders, so it was in the lab, so it is in the field, a mixed composition outperforms either monotypic composition. So it's good to have a mixture of personality types when you're not interacting with a heterospecific. You throw 1 foreign spider into the group, and presto changeo. That positive effect of within-group behavioral diversity vanishes. So this foreign spider has a negative impact on the performance of all aggressive individuals, has a negative impact on the performance of a mixed society, and has no effect on a colony of all docile individuals. And that's where this foreign spider is actually gaining the most mass. I thought, or I presumed, because they were exploiting these docile individuals, but they're actually having no negative impact. But you might say, well, the docile females were doing pathetic before, they're doing pathetic afterwards. There's only so bad you can do, right? Well, if you throw in a Lerinioides cornutus, it has a negative impact on a colony of all aggressive females, has a negative impact on a colony of mixed composition, and has a positive impact on a group of all docile females. And what that means is that whether or not a foreign spider is a friend or foe changes in direction and magnitude based on the personality type of an individual spider and the personality of your neighbors, right? And if you were a community ecologist going around just looking at the effects of species A on species B, data like this should kind of shock you a little bit because it would suggest that you couldn't even predict the kind of species interactions that an individual is engaged in if you failed to account for individual variation. And here, it's not individual variation in color or variation in body morphology or caste or something. It's a cryptic kind of diversity that is invisible to you unless you put your animals into a scenario where they can express the variation. And I can't go around to you saying, you, I don't know, coral reef biologist in Moorea, you should go around and measure the personality type of every fish on the reef. That would be imbecilic, impossible, and I can't recommend it to you. But what I would say is that data like this should at least let you have a pause for a moment where you think about whether or not there's some key players in a community that you might begin to open the door to evaluating individual variation in those species and see what it buys you. And if it's nothing, just close the door and forget about it. But I'm actually shocked when community ecologists don't account for individual variation and still get significant results. My breath is taken away. All right, so if you look at the relationships between these foreign spiders and the foundation species, there are mensalisms when a colony is composed of all aggressive individuals. There are mensalisms when they're composed of a mixed composition. And then all those mensalisms turn into either commensalisms or mutualisms in an all docile society. And so the very network of species interactions that an individual or a group is engaged in changes radically based on subtle shifts or radical shifts in a cryptic element of trait diversity. All right, so if you, I don't know, maybe if R squareds matter to you, our ability to predict the performance of both the foundation species and the foreign species goes from being able to explain about 15% of the variation, if we just account for species ID, to like 50 or 60% of the variation in a highly contrived experimental framework, but 50 to 60% of the variation when we account for individual variation in behavior. So if it's our goal to make ecology a more predictive science, and I would argue that is all of your goal and mine too, then I think one of the ways that we might do this is trying to account for variation in key functional traits, which might not be behavior in your system, but it might be something that's invisible to you otherwise. So that's my finger wagging portion of the talk. It's over now. All right, so if we manipulate the ratios of types of individuals inside of colonies, we change the magnitude of species interactions, we change the directions of species interactions, and that's very satisfying to me and quite unexpected. I've given this talk a lot of times, and yet I still find myself getting excited about those data, which means they can't be that bad. Or I'm really good at self-trickery. All right, so all of the experiments up until now are sort of cutesy and short-term and involve manipulating little ratios of individuals and looking at things over a series of weeks in kind of like a mesocosm-like framework. But my favorite papers in ecology actually follow things for years in the field and try to get at the long-term story. And I think this is also really important for when you give job talks, that you have some really heavy-hitting big papers. And I wanted to try to get at those when I was looking for a job one day. So the next few talks, next two components of this talk will be long-term studies that persist for years. And we'll talk about the performance of whole lineages of colonies and not just a few individual spiders. So the question here is, can the personality types of the founding members of a new subpopulation predict lineage performance generations downstream? So if a new subpopulation is founded by docile individuals or aggressive individuals, does that change the probability of whether or not the descendant lineage is still around years later? How rapidly it's proliferating through space and its general stability. And so the reason you can evaluate this in analysis with studiosis is that on average individuals have a low dispersal distance. 95% of individuals disperse within 1 meter of their parental colony when they leave, and then 99% within 2 meters. 1% go very, very far and possibly die. We don't know what happens to them, but anyway. But in general, you can set up an experimental colony, and if you remove all of the non-focal colonies, you can watch descendant colonies emerging in the immediate vicinity of the progenitor colony for generation after generation in real time. And if that's too hard for you, when these spiders disperse, they release a little thread of dragline silk that's highlighted in the morning dew or the evening sun. So if you go out in your kayak each day and go check on these colonies when they're dispersing at a characteristic time early on in the summer, you can actually see a highlighted thread of how they got to their new spot on the tree, right? Yeah, it can't possibly be, I mean, it's not that easy. You're just like kayaking around. I'm making it sound like it's super duper easy, but it's at least possible. And for most systems, it isn't. And then founders are key. Oh, so a couple of things. Colony extinctions are a neighborhood phenomena. So I mentioned that a lot of colonies go extinct. It's not just one colony here and one colony there. No. It's a neighborhood phenomena. All of the colonies in this area die together. And so there's the possibility that if a progenitor begets a really successful lineage, that whole lineage could still die together in unison as a neighborhood extinction phenomena. And founders are key for a few reasons. One, if their traits are genetically determined, the degree to which they are, they determine the genetic milieu that's available for the population and for selection in the immediate future. And then secondarily, whatever they do to the environment, however they manipulate the environment, that altered environment is inherited by their descendants in subsequent generations. And both of those elements become key in this talk. All right, so the hypothesis here is that aggressive individuals will act as a kind of social immune system, keeping these foreign spiders at bay, at low enough representation, that the whole lineage should do better by having them around. But that should be costly, right? If you're defending your colony from voracious predators, presumably that costs you energy and possibly fecundity or survival. All right, so what did I do? I staged founder events of either 2 docile females, 1 docile female, 1 aggressive, or 2 aggressives. And then I tracked lineage vitals, basically, for generations downstream. How many new colonies are being formed, offspring colonies are being formed each year, the incidence of lineage-wide extinction events. When I say lineage-wide extinction, if there's 1 descendant colony still around, I count the lineage as still being alive. So when I say lineage-wide extinction, all the progenitors and all of the descendants in the immediate vicinity are gone. The number of inquilines inside of each of the colonies, and then lineage dispersion. How far away are the descendant colonies being founded from the progenitor? And I have two treatment groups. I either allow inquilines to enter all of the colonies, or I remove the inquilines just from the progenitor colony. So the inquilines can still infiltrate everyone else, but just that progenitor colony, we remove inquilines from there. This goes up to 2013 in this talk, so it's for 6 years. So, are the traits of lineage founders associated with lineage productivity generations downstream when inquilines are present in all of the colonies or when inquilines are absent just from the progenitor colonies? The y-axis here is the number of new offspring colonies produced each year when you're founded by a group of docile females, a mixed phenotype composition, or all aggressive. Well, when you have when your founders are all docile, lineages founded by docile individuals are far more productive. They're producing more offspring colonies each and every year than rival lineages founded by mixed compositions or aggressive individuals. So they're more productive. They're sending out more offspring. When you remove these inquilines, the rank order totally changes. So if you remove the inquilines just from the progenitor colony, suddenly the most productive lineages become the least productive lineages. And then the least productive lineage becomes intermediate. So the rank order performance of rival lineages in space is dictated by the traits of the individuals that founded them generations before. And manipulating key selection pressures like these inquilines radically alters the rank order performance of rival lineages. But in real life, inquilines can enter all of these colonies, right? So why isn't the whole world overrun with docile lineages? Because they are so productive. Well, if you look at lineage persistence through time, that sheds some light on it. We're looking for lineages of all docile individuals in light gray, mixed in dark gray, and all aggressive in black. And we're looking at year census along the x-axis. The y-axis is the proportion or the number of lineages that are still present, that at least have one offspring colony still around when inquilines can enter every colony or when they're removed from the progenitor colony. So yeah, docile lineages might be the most productive. They're spewing out offspring colonies, but that's a short-term advantage. Because it turns out, even though they're sending out all these daughter colonies, it's very likely that the progenitor colony and all of its descendants are going to die within just a few years. And actually, we've followed these colonies out until 2015, in which now they're finally all dead. So the least productive lineages, the ones that seem to be the most sickly in terms of the number of offspring colonies they produce, are much more likely to persist through time. And if you remove the inquilines, all of the lineages have a very high probability of persisting through time. And this sheds light on the idea that these foreign spiders are a major driver of lineage-wide extinctions, and that the most productive lineages might not be the most successful in the long run. It might be an evolutionary dead end on an eco-evolutionary timescale. So, how do lineage extinction events happen? The first colony to go extinct, 94% of the time, is the progenitor. When the progenitor colony dies, the inquilines, they don't die. They're fine. They're normal spiders. They have a full-time job living on their own. They're totally fine. They don't die. They just leave. And where do you think they go? They move to the suburbs, right? So they move to the suburbs, and they infect these incipient tiny colonies that barely have any females in them at all, and now they have a wave of foreign spiders that killed the progenitor. And that pulse quickly drives the extinction of those descendants. And in real time, within a few days, you can watch the very same inquilines that killed one colony move to a smaller colony and kill them too. A little hopscotch of death in situ. So more diffuse lineages are less vulnerable. So if descendants are moving further away, presumably it's harder for the inquilines to find them, and it's more likely that at least one of them is going to live. And that alone explains 29% of the variation in individual survivorship. And then this thing here, so we're looking at, when we individually marked some of the inquilines and some of the progenitors, and we looked at what proportion of them showed up in a descendant colony that year. So 80% of the inquilines that we marked inside of a docile progenitor showed up in a descendant colony later that year. And only about 30 or 40% of inquilines were inherited by a descendant in aggressive lineages. And there's a couple of reasons for this. One, when offspring colonies are founded in docile lineages, they're founded on average 15 centimeters away, right? So it's a new web, but it's right next door. And they have that little highlighted route I talked about. Presumably the foreign spiders don't have a hard time finding them. Versus it's on average 40 centimeters for a lineage of all aggressive individuals. So the difference between 15 centimeters and 40 centimeters seems maybe small, but I think it's biologically significant. And then once these inquilines find a descendant in a docile lineage, they're docile. They're not going to chase them away. Versus the descendants of aggressive colonies are presumably, on average, more aggressive, which means they're potentially better defended as well. All right, so the founders, personalities of founding members of a population can shape lineage productivity and extinction risk generations later. Their effects echo through time. There's a trade-off between the productivity of a lineage, how rapidly it grows and sends out offspring colonies, and its susceptibility to social parasites, and then extinction. BTs just means behavioral types here. Think of it just like as personality types. And then lineage extinction is caused by inheriting the progenitor's parasites. The same thing that killed your parents killed you eventually. And the reason that I think docile lineages are so susceptible is because the progenitor colony gets so many of these foreign spiders so quickly because it's never defended, right? And then that pulse of foreign spiders is what drives the extinction of the whole lineage. I think it's pretty significant, or I like to think that it's not trivial. All right, so, and if you think about this, it's kind of like R versus K selected species, right? So there are lineages that as a consequence of their progenitor traits grow very rapidly but are more susceptible to lineage-wide extinction. And then there are other lineages that are slow growing and apparently more stable. I'm not going to say that they're adapted to those sorts of selection pressures, but it does at least bear resemblance to some of the trade-offs that are experienced by individual level organisms that are well known to us. And it's possible that there are strategies of honeybee colonies that grow rapidly and are more susceptible to mites or whatever, and it's and slow-growing honeybee colonies that are less susceptible. It's possible that trade-offs could be occurring anywhere in all sorts of social groups, but you would never know, right? It's impossible to staple together a bunch of lemurs, leave them for multiple generations in the field, and track them and over the course of your lifetime. It's impossible, it's unethical, et cetera, but I at least want to open the door conceptually to the possibility that it might be happening, and it's difficult to ignore the intriguing notion, for me anyway. All right, so has any of this selection caused any real adaptation, right? So we know that there's lots of relationships between these compositions and various sorts of outcomes. So the question is there a right mixture? In the face of all these selection pressures, is there an ideal mixture of docile versus aggressive individuals? And if there is or there isn't, can you even achieve it, right? And how would you even maintain such a composition? Do you incentivize sticking around for socially influential individuals? Can you fire losers from your colony? We have HR departments for trying to manage such things inside of human societies, but I mean, it's for an animal social group, it's quite complicated. And I think that all collective phenotypes are actually kind of a weird thing to have evolve, especially in a diffuse society like cooperative breeders or societies where everyone's reproductively totipotent, because each and every one of these agents could mess up the composition of the group just by reproducing, right? So we like to think about collective phenotypes as being very highly organized and very nice, but actually equally as likely we have like beached whales and riots of people, so sometimes collective traits are not so good. Anyway, so along the distribution of this species from Maine to Patagonia and Argentina, environments vary a lot. And some of the environments that we focus on are immediately below dams, and they're high resource environments which have a lot of dissolved oxygen because the water is quite cold when it's pulled out beneath a dam. So there's high prey availability from aquatic insects that pulse out in the spring each year. There are typically more of these foreign spiders at those sites because they're high productivity, and disease outbreaks are more common. Far away from those dams where dissolved oxygen is lower and eutrophication is more common, prey availability goes down, social parasites, foreign spiders are along the riparian environment, are less abundant, and disease outbreaks are rare, which are the classic high versus low resource site constellation of selection pressures. Interestingly, there are also stable differences in colony compositions in high versus low resource sites. Along the x-axis is colony size, all the way to the left would be a small colony, all the way to the right would be a large colony, and the y-axis here is the proportion of colony members that are the aggressive phenotype. So towards the bottom would be a colony of all docile individuals, to the top would be a colony of all aggressive individuals. At high resource sites, small colonies, mostly docile. Large colonies, mostly aggressive. Low resource sites, small colonies, and I know this is not linear, small colonies, almost all aggressive, large colonies, almost all docile. So there's site-specific relationships between colony size and composition that are at least associated with what could be different selection pressures, suggesting or hinting at the possibility that group composition could be an adaptable trait, which has been hinted at in the literature for like 50 years, but the tests have been really equivocal. This is not my hypothesis. This was articulated a long time ago. So the hypothesis here for us is that colony composition could be locally adapted as a consequence of site-specific colony selection as mediated by colony extinction events. But this hypothesis was first articulated in terms of caste ratios that could be locally adapted in social insects. For me, I just cut out caste, put in personality, and then evaluated the same hypothesis. So what did I do? I created 53 experimental colonies per site and deployed them out at each site with random combinations of size and compositions. 37 of these colonies at each site were composed of native individuals. I took them from a site, I messed with their composition, I threw them right back out to the site where they came from, where they have an evolutionary history. 16 of these colonies, I took individuals from the field, I messed with their composition in some way, and then I threw them out into an environment where they don't share an evolutionary history, from high resources to low resources and vice versa. I then tracked colony performance in the wild, whether or not they live or die for the next 2 generations, and I tracked the number of social parasites inside of the colony, the proportion of egg cases that are being cannibalized by the social spiders themselves, and whether or not you live or die. And provided you live, I messed with your composition, which might be adapted, and then I throw you back out into the field and you manage to survive. Can you fix your composition, if there is such a thing? And can you only do that at some sites versus others? All right, so I know it's an unattractive figure. But on the left-hand side here is the naturally occurring relationship between colony size and composition. This is the starting relationship between colony size and composition at each of our sites for high resource sites. And as you can see, there's a naturally occurring relationship that's positive. The starting relationship is flat. And these are the colonies that survived 2 generations of selection in the wild. So it's only colonies that follow along that positive relationship that stick around, right? So only the compositions that resembled naturally occurring compositions, and it's a combination of size and composition, are the ones that persisted, which means there's a right mixture to have. It changes as colonies grow, and naturally occurring colonies are good at achieving it, either because the ones that didn't died, or they have regulatory mechanisms that allow them to deploy their optimal composition. Low resource sites, the naturally occurring relationship is in the opposite direction. Starting relationships are flat as before. It's the same combination at each site. And these are the surviving colonies. So here, again, it's only colonies that follow along that negative relationship for low resource sites that persist through time. So only colonies that resemble the native compositions survive. Again, suggesting that native colonies can either have some way of adaptively regulating their composition as they grow, or the ones that stray too far from the ideal mixture, those ideal combinations, perish. All right, so why? So why is it that at some sites a composition can help you, but at other sites the same composition kills you? Well, the correlates of extinction are totally different across these sites, right? So green panels are the high resource sites, yellow panels are the low resource sites. Colonies that survived are in black. Colonies that went extinct are in gray. Top panels are the representation of these social parasites. Bottom panels are the proportion of egg cases that were cannibalized. At high resource sites, the colonies that went extinct had tons of social parasites. Colonies that survived didn't. There's no relationship between egg case cannibalism and persistence at high resource sites. Low resource sites, social parasites, there aren't a lot of them anyway. They're not associated with extinction. There, it's all about egg case cannibalism. Your sisters eating your egg cases is what drives you extinct there. And presumably these different selection pressures on colony extinction change the ideal mixture and beget those site-specific relationships that we see in situ. But can a colony do anything about a bad mixture? Is it just a glass cannon? If you just happen to fall into the right mixture, you're going to live, and if you don't, you die? Or can a colony fix something if there's a bad mixture there? So the top panels are high resource sites, the bottom panels are low resource sites. Foreign colonies, the ones that moved across sites, are in black. Native colonies that persisted at their native sites are in gray. And then the y-axis here is the change in colonies' distance in phenotypic space from the ideal mixture at their current site. So negative values means that over time, if I mess up your composition, you can walk your way back towards the ideal mixture at that site. Positive values means that if I mess with your mixture, you're walking your way further and further away from the ideal mixture at that site. If you're a native colony, you're still at your site of origin, you can fix your composition slowly if I mess with it. But a foreign colony displaced into an environment where it shares no evolutionary history, even after several generations, is just getting further and further away from their ideal mixture. Maybe it's because foreign spiders are stressed. Maybe it's because they're dumb. Maybe it's because they lack the necessary plasticity. Or maybe it's because they're doing something else that's totally adaptive and makes sense. It just makes no sense in this scenario. And that is that they're moving themselves closer and closer to the ideal mixture of their site of origin. So even though they're at this new site for multiple generations, they're still recapitulating the ideal mixture that would have been advantageous had they still been at home. And this provides evidence that these regulatory mechanisms are site-specific and locally adapted to help colonies circumvent extinction and the individuals within them. We're not talking about altruism here. It's good for everyone to live. Take-home messages. So group selection on colony composition is different between high and low resource sites. There's an ideal mixture to have. It differs across sites. The correlates of extinction are also different between those sites, and that might have some explanatory value for why the ideal mixture changes across environments. And colonies appear to have responded to this, and individuals within them have responded to this by evolving mechanisms of regulating group composition to help your kin and colony circumvent would-be extinction. So let's say that you think behavioral ecology is stupid. You think behavioral ecology, a bunch of 1980s mechanistic minutia that wore out its welcome years ago, and is ecological stamp collecting to the worst degree, a phenomenological snake eating its own tail. Well then, to you, I would say that, all right, fine, whatever, I'm not going to argue with you. And you can reparameterize what I do, and actually what a lot of people do in other terms. And in this case, we're manipulating individual variation in a foundation species and observing the effects that it has on intrinsic growth rate, population stability, the network of species interactions that occurs in groups, and then population and community stability in ways that I don't think most people would think were possible using a trait-based approach, which is the holy grail for some ecologists, trying to find significant relationships between traits and higher order ecological outcomes across environments. And with that, I'd like to acknowledge Susan Reichert and Andy C. for tolerating my frenetic, high-energy behavior for years upon years. Jennifer Fuel and Stephen Pratt for basically getting me down this pipeline of thinking about group composition as something that could evolve in response to selection. And that literature was already present and quite deep within the social insect literature. Lena Grinstol, Virginia Setapani, and my graduate student Carl Nick Kaiser helping me collect a lot of these data. Additional field assistance from Thomas Jones from East Tennessee State University, Jessica Purcell from UC Riverside, who's now in your backyard. You should totally invite her out if you haven't had her here yet. And then funding from the National Science Foundation, the Binational Science Foundation with Israel, and most recently an NIH grant, which I'm actually totally shocked about, but God bless them too. And then finally, I'd like to thank a suite of collaborators, consultants, and friends here and all over the world that remind me that science is a dialogue, and thankfully none of us are in it alone. And thank you very much for listening. Got some questions.
I'll let you pick up.
This one right here, and then you, Bjorn.
So I missed something about the mechanism involved. So your docile colonies either aren't negatively affected or positively affected by the presence of these heterozygotes.
Yep.
Right? But then you showed that those same docile colonies go, the lineages go rapidly only in the presence of these heterozygotes and not in their absence.
Oh yeah. No. Well, so I have I have data that bears some light on this, but I'm not totally enchanted with it yet. First, I think that the effects of these foreign spiders on docile colonies are dose dependent. If you have one or two foreign spiders, maybe you can do okay. Maybe you can deploy some sort of strategy. But as these foreign spiders become, enormously common inside of a colony, then that strategy falls apart. We have a cutesy little study that's kind of contrived that looked at pairwise interactions between docile females and heterospecifics and aggressive females and heterospecifics. And so and it's sort of like a producer-scrounger game. With an aggressive female, they're more likely to capture prey, and then the individual that joins them is the heterospecific and ends up chasing away the aggressive female. When there's a docile female involved, the spider that tends to capture the prey is the heterospecific, and then docile females basically just wander over on the opposite side of the prey item and co-feed with them, and it's sort of like a kleptoparasitic strategy. And I should say that that's the same thing that docile females do with aggressive conspecifics. So when there's a mixture of a mixed composition inside of the social spider society, the first individual to attack is usually aggressive, and then docile females sort of like slowly walk over and co-feed with them. So I think it just happens to be that docile females have a strategy that works with both aggressive conspecifics and heterospecifics when they're at relatively low representation within the group. But I'm still not totally sated with that response. I mean, it was cutesy, but it's all highly contrived. And actually, mechanisms are hard. But that's the best I got right now.
Okay.
Go on.
So you highlighted a number of factors that most likely explain the outcomes of these interactions among different conspecifics and non-conspecifics.
Yeah.
But if you were to imagine modeling such a system to a degree that where everybody would agree that it has a high explanatory power,
yes.
Do you think that there are factors that you don't know about, no one knows about, that are important?
Yeah, well, I mean, I've sort of taken like a candidate gene approach to like in a behavioral landscape to this system. So very early on, I identified one behavioral access that was very quick to be able to analyze, to assess, and that had a high level of predictive value for various sorts of outcomes here. But and so I've sort of hyper focused on that. But there probably are all sorts of other behavioral axes and other axes that are yet unidentified that could be important here. Are aggressive females at one site the same kind of aggressive females as aggressive females at other sites, or could they differ at other behavioral axes? Absolutely. clearly, I mean, and all of this is still sort of conducted in a highly controlled experimental framework. And so our ability to predict what happens in wild colonies using these same sorts of variables, so sort of a correlative observational approach, we can only explain about 30 or 35% of the variation in colony performance in different sites. So clearly, in real colonies, in real life, there are other factors going on there too. But I mean, these are at least promising leads. I should also say the National Science Foundation is not interested in funding any of this work any longer, no matter what I do. So now they're interested in funding stuff that I know nothing about, because evidently that sounds much more exciting. But we've pivoted into another system. But I think there's a lot more that can be done here. If you're an interested graduate student, you should come talk to me for a PhD.
Yeah, for some obscure reason, I'm interested in finding a role for males.
Yes. All right, sure. No. What am I?
Are males docile and aggressive?
Yes.
Is there any behavioral sort of assortative mating across males and females?
Right.
On their personality?
Yeah, so males do have docile aggressive distinction, but it's not bimodally distributed. So if you look at the distribution of inter-individual distance scores in females, I think, oh yes, there's an aggressive mode and a docile mode for females. But for males, it's unimodal, but their phenotype does help predict the distribution of phenotypes in offspring. So there's clearly something to it. More aggressive males tend to be slightly larger. They're like 4% larger than docile males. They're more likely to win access to females, both docile and aggressive, but they're also more likely to be killed off by aggressive females than docile males are. And so we have, again, these sort of like cutesy behavioral assays where we allowed one aggressive male, one docile male to compete over a docile female or an aggressive female. And we found that almost universally, aggressive males win access to the female, but they're much more likely to be killed off by aggressive females. And so literally while the aggressive female is eating the aggressive male, the docile male will go over and mate with her. And so I think we called it something really cutesy, like to win the battle but lose the war, whatever the hell that was. But basically we think that the available evidence now suggests that it might be discriminating, but in situ we have no idea. But there's probably something there. Here.
I was wondering if you find like these two stark contrasts of personality, or if it's rather a continuum of like ranging from aggressiveness to docileness.
Yep. For this axis, it's two modes for this species, but I looked at 16 different species of Anolosomus that vary whether they're subsocial, which is just a single female and her brood, or social, which has multi-female colonies, and sociality has evolved independently in this genus at least 6 times. So if you look at the distribution of subsocial species, it's unimodal, and all of the social species have bimodal. And so it suggests that with either the evolution of sociality, you get a bifurcation in those personality types associated with some sort of social organizational element, or that over the course of individuals' lifetimes, they experience plasticity, they bifurcate out in behavioral niche space, or something like that. So it's not an artifact of how we test this. Subsocial species actually have a unimodal distribution, and there is an increase in behavioral variance across many axes for social species.
And how long do the individuals live?
About a year. Yeah, and they're all born at the same time, and then they all.
Chances that their personality type changes over
Ah.
years, like
Yeah, so the repeatabilities aren't one, right? And individuals, you can starve individuals enough or heat them up enough that docile individuals will become more aggressive. But at least with heat, we've done experiments where when you ramp up the heat, their aggressiveness increases, but they maintain rank order. So the docile individuals become more aggressive, and the aggressive individuals become more aggressive. It's hard to test in this situation because the box is only so large. But if you look at latency to attack or activity level or things like that, they maintain their rank order differences. But if we starve females, we can get a shift in the representation of her offspring that persists throughout their development. So it does still respond to plasticity, or it does still respond to the environment.
Okay.
All right, thanks so much. Thanks for having me. Bye.